Hello, everyone. I would like to welcome those participants who have just joined. We will be starting in more 5 minutes from now. We are waiting for everyone else to join as well. And meanwhile, we begin with today's session. It would be nice if you can put in the chat box about yourself. It could include your educational background and also something about your research interest. Thank you. Hello, everyone. I would like to welcome you all for the first session of Genomics Data Analysis. This is an online bioinformatics training program that will provide an in depth overview of concepts and analytical tools used to study genomic variations. The program is designed to introduce students, clinicians, and researchers to the and the applications of genomic data analysis to study genomic variation and apply genomics in translational sciences to improve diagnostic accuracy, predict which drugs are likely to be effective in patients, and also contribute to the monitoring, treatment, and diseases in individuals and populations. Today, we will discuss about the course structure, expectations, and the intended outcomes of this particular program. We will discuss about the applications of bioinformatics and also discuss about few of the coursework that you can definitely go through, including introduction to bioinformatics. We will discuss about examples in biomedical research, pharma R&D, and agriculture as well. Each week, participants will meet online to discuss various aspects of NGS data processing, genomic data analysis, and interpretation. After the first introductory lectures, participants will have the opportunity to get guidance and also work on curated datasets or develop independent projects that leverage the tools and concepts covered in this program. I would first of all like to introduce myself. My name is Sunalika Ray, and I'm the Omics Logic Community Manager. And I'm responsible for daily interaction with thousands of users that are a part of our bioinformatics community that is managed by Pine Biotech. Omics Logic programs are a result of close collaboration between Pine Biotech, an innovative big data analytics company which is based in the US, and numerous academic institutions that have participated in content evaluation, curriculum review, and project design. And as a result, the Omics Logic programs have been thoroughly tested around the world and are known to lead to excellent outcomes for students with minimal bioinformatics background, including biology and biomedical study majors, as well as faculty interested in bioinformatics, data science, and biomedical research. So, before we start with today's session, I would like to introduce us. As already told, we are a US based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy to use analytical tools. And our mission is to make bioinformatics more accessible. Our training programs are running in five different regions with over 15,000 plus users around the world who have completed the training programs. The programs follow a project based learning approach using research grade tools to analyze data from top peer reviewed journals. The training has been completed by 15,000 participants from 162 countries in over 300 workshops for six different specialization tracks, which include oncology, infectious diseases, precision medicine, neuroscience, data science for biomedical data, and comprehensive training on omics data analysis. Over the past several years, we have had the privilege of working with a number of organizations developing a training program for research oriented students, faculty, and democratizing bioinformatics, making this discipline accessible to thousands of students and faculty around the world. We will combine online sessions, self guided training, study materials, and practical assignments as well for an immersive experience that has proven to be effective in these training programs, which are known as Omics Logic. I take the honor to introduce the founders and the team members of the company. Dr. Alfred Tauber comes from a medical background with an expertise in oncology, immunology, and biochemistry of information. And one of his major projects is the establishment of Bioinformatics Research Center, the Tauber Center, that has been at the core of what we do. And throughout the program, you will get the guidance and go through the server as well. Dr. Leonid Brodsky is the director of the Tauber Bioinformatics Research Center and is an expert in bioinformatics and biostatistics. Our platform is a result of hard work by some of the people who are mentioned here on the right. Julia Panov is an expert bioinformatician. Avi Titavsky leads the development of the platform. Dr. Harpreet has an expertise in the application of statistical machine learning techniques and biomedical data and looks after the curriculum development. Dr. Mohit Mazumdar is an experienced bioinformatician and has multiple publications in peer-reviewed journals. He has been developing collaborations with universities as a mentor and project lead for Omics Logic Training and Research Fellowship. Dr. Raghavendran is an accomplished computational biologist with an experience in large-scale analysis of multi-omics data and data science methods in precision oncology and protein DNA structural bioinformatics. We will today also hear from Dr. Raghavendran and Mr. Ilya Brodsky. So let's start by signing up on the learn.omics logic portal. This educational portal will help you get started on your bioinformatics journey, and also throughout the course, you will be given directions to access the T-Bioinfo platform for bioinformatics processing and analysis of transcriptomics data. The platform includes demo pipelines as well as data management and analysis cloud infrastructure to run bioinformatics pipeline. The different stages of analysis are performed in different sections of this multi-omics platform. Now, to proceed with, before we go ahead and sign up on the Omics Logic Learn portal, I just quickly like to launch a poll so that we can know the audience who have joined this particular session and also get some knowledge about their backgrounds. Dr. Mohit, can you launch the poll for me? I can see you have also joined the session. And meanwhile, let me share my screen. Okay, so I am now going to put the link of the portal in the chat box. Thank you so much, Dr. Mohit. So my first question is, what is your educational background? Are you a graduate or a postgraduate or a person with a background in PhD or postdoc? And my second question is to know about your research interest. So what is your research interest? Is it infectious diseases, neuroscience, precision medicine, agriculture, or space omics? And my third question is, what outcome do you seek after joining the program? Is it a specialized training that you want to go through or a research project? So we'll have this poll up for more 30 seconds, and it would be nice if you all could answer the poll. And for those who have answered the poll, I'm sending in the link to learn.omixlogic portal in the chat box. If you have already signed up on the portal, then please drop a 1 or yes in the chat box for our knowledge. And if you are new and joined the session for the very first time and have not yet signed up, then please go to the link and let's create an account for yourself. So I'll just do a quick demo so that you can see how you have to sign up. Just click on the login button on the top right, and this will redirect you to an interface like this. Now you either click on create an account for yourself, put your mail address, create a password for yourself, put your details and create an account. Or you can also create an account by simply using any of your Google accounts or Facebook accounts or other social media accounts. So I'm awaiting the responses in the chat box. Thank you, Karl Marx. Those who have signed up, please let me know in the chat box so that we can proceed further. Thank you, Surbhi, Bhupender. Yes, so after answering the poll ratio, it would be nice if you could go onto the portal, that is learn.omixlogic.com, and create an account for yourself, because this will help you go through the resources, various resources that we are going to discuss today, and work with them. Okay, so I can see a lot of you have already signed up and are signing up. Those who have completed their sign up, the next step for them is to complete their profile. So now to complete your profile, you need to click on this mail address of yours in the menu bar, and this will redirect you to the profile section. You need to ensure that you have an updated profile picture. Your name is in the title case, because for each and every coursework that you complete on the portal, you'll be given a certificate, and the certificate will have the same name as you type in your profile. So please make sure that your name is not in capitalized format or small alphabets. It should be in title case. Also, please link in your social media accounts so that we can feature your profile based on your progress, and also add a brief bio about yourself. That could include your educational background, your research interest, what you want to work on, etc. Also, for every coursework that you complete, you will get certain points. And these points are going to take you from a beginner level to an expert level. So you can see your courses that you complete, your progress here in the profile tab with the points that you get on the achievements that you have completed. And also those who have already joined this particular program, Genomics Data Analysis, please make sure that you have the badge of genomics in your profile. This will ensure that you have the access to the program page. I'll just shortly take you to the program page and show you the relevance of the same. Also, you need to ensure that you have a starter subscription active that will give you an access to all the courseworks on the portal. And if you don't have the badge or the starter subscription, then you please need to tell me so that I can give you the same. Okay, so now coming to the program page. So this is the program page of the program for which we are here today, that is genomics data analysis. Now, on this program page, you will find the session schedule of the program where you can see, first of all, the days for which each and every session has been scheduled. Also, the topics that will be covered in each of these dates and the associated online resources. Now, with each and every session, you need to go through some resources that we have mentioned here. You need to, these all resources are hyperlinked. As a part of the session completion, you have to go through these lessons as well and make sure that you complete them before you actually click on the complete session icon that will come up here after each session. I'm going to upload the recording of each and every session as we completed here on the program page itself. So to revise the session or go through it once again, or let's say you could not join the live session, you were busy somewhere else, you can definitely refer to the program page and go through the recording at your own pace and complete the resources as well. And then you just click on the complete session icon. It's very important that you all, the program participants, should have the access to the program page, because while completing each and every session that is mentioned here on the program page, after you do so for all the sessions, you will be able to get your certificate towards the end of the program, towards the completion of the program. So that was the relevance of the program page. And before we actually get started with today's session, I'll just take 2 more minutes and assist you all on how you can navigate through the various courses that are present on the learn.omixlogic portal. And then we'll hear from Mr. Ilya Brodsky and Dr. Raghav and get started with the session. So once you are here on the portal, you'll see various categories here. So if you click on the courses tab in the menu bar, this will redirect you to all the coursework that is there on the portal. Now coming to the section of tags that you can see here on the right side, just by clicking on the tags, you can filter out which category you would like to explore and learn more about. So if you just click on course, all the courses that we have on the portal will just come up and you can go through them and explore more. Now, throughout the program, we are going to give you directions on like which and how, which particular lesson you have to go through, and what resources you have to go through, and what all exactly you have to practice and get some hands-on. So I'm just giving you a rough idea of how you can navigate on the portal. We'll be giving you more detailed ideas when we just go along each and every session. And if you want to have a look at the projects, now these are some example projects which will help you understand the applications of a particular key concept, let's say, that you went through in the coursework, and will help you understand how the concepts that you learn in a particular coursework could be applied to answer a particular research problem or a research question in hand. So these are the list of example projects that you can explore and learn from. Also, then you have various specialization fields that you can specifically pick and go ahead. So if you just click on project and then click on genomics, so you will get all the example projects that are related to the specialized field of genomics. And yes, then definitely you can pick the field and explore more. So that was the easiest way of how you can navigate on the portal. So now I would like to pass on this session to Mr. Ilya Brodsky to let us get started with the session and learn more about genomic data analysis and why is it important and what is the relevance. So over to you, Mr. Ilya.
So Nanika, please make me co-host so I can share my screen.
Yes, Ilya, you are a co-host now. Thank you.
All right, so great to have all of you join today. I'm going to briefly cover some of the topics that we're going to be discussing for this program on genomic data analysis. So the objective of this program is really to cover a broad overview of different types of topics, provide you with the methods that you can practically apply to analyze the data, genomic sequencing data, but also importantly to understand how this can be applied to a variety of different project examples and try it out on your own. So hopefully by the time that you complete this program, you will understand what the terminology means, especially when you read the papers. You will also be able to practically have experience and know where to start your project, how the different stages are connected to each other, how to interpret this, and have a meaningful discussion with your peers or with others about genomic data analysis and interpretation. But today I want to briefly start from the very beginning. Later on, I will show you where you can find a lot of the information that we cover in today's session. But I will just take a few minutes to talk about some of the basics, because we typically assume that everybody's on the same page, but a lot of people, just by not understanding certain types of terms or maybe concepts, it's very difficult to follow along. So what is DNA code? What do we know about the DNA code? And what is it that we still don't know? What is there else that we are trying to learn? Why do people continue to study genomic data and try to interpret and understand what this code means? So DNA code, especially when we talk about eukaryotes and we talk about people, we think of this code as a book of recipes. So it's organized in a specific structure. It has sentences, paragraphs. Paragraphs are organized into chapters. And so really learning this book and how to navigate and spot these patterns is the basic approach of genomics. So DNA is short for deoxyribonucleic acid. It's a long molecule that's composed of two chains that coil around each other and form this double helix that we all know. And the code contains instructions. So these instructions are coded in the human genome in about 3 billion base pairs. So it's very long. Not all of those base pairs contain information that ends up being translated into proteins. And similar approaches of putting information into this code are used by bacteria, viruses, and large organisms, multicellular organisms as well. So the same approach, the same code contains directions or recipes for what we call life. And life is the condition that, that distinguishes animals and plants from inorganic matter. So what does life give us? It gives us the capacity for growth, reproduction, functional activity, and continual change that precedes death. So when we talk about life here, you can see a really nice animation, or actually not an animation, a video, where a single, small microorganism, and maybe even different kinds of microorganisms, are moving, right? And that's really what distinguishes life from these inorganic matter, like rocks, for example. So when we have these, these recipes or these pieces of code, they are organized in a very tight way by compressing this long molecule into a very tightly compacted nucleus. And when cells divide, what happens is that in the nucleus, we have all of this DNA, and when it divides, we can see how this DNA is organized because it needs to be unfolded. And so what we discover are these strings that are inside, and we can actually see them. So this is the process of division, and what you can see are these chromosomes, right? So these chromosomes are actually, string of DNA, also packaged very tightly in these, specific shapes. So these chromosomes contain different genes. Each one of our cells contains 23 pairs of chromosomes. One of the chromosomes comes from our mother and the other from our father. And the unique structure of chromosomes keeps DNA tightly wrapped around spool-like proteins called histones. Without such packaging, DNA molecules would be too long to fit inside our cells. So when we think of this chromosome, we can expand it, and this is what it looks like. So here's the actual DNA, this string right here that is coiled around these histones. So if we continue and unfold it, you can see that these are the histones, and it's packaged right around those histones. So when we speak about this code and what does this code actually contain, what instructions does it contain? So the product of the code and the way that it interacts with the environment is called a phenotype. So a phenotype can describe a single cell, a tissue, organ, or the whole organism. Phenotype of an organism might include height, size, and activity, and phenotype of a cell might include its specific function, size, and morphology, or its shape. So the code, which is packaged in this molecule, defines this phenotype, right? Defines first phenotype of the cell, then of the organ or the tissue, and then of the whole organism, right? And so the basis of this code is all about these nucleotides. So these nucleotides are made up of a sugar called deoxyribose, a nitrogen-containing base, and a phosphate group. The nucleotides are joined to one another in a chain by covalent bonds that form between the sugar of one nucleotide and the phosphate of the next. And the result of this structure is an alternating sugar-phosphate backbone. So each strand of DNA molecule consists of a sequence of bases that is complementary to the bases in opposing strand. So when we say base pair, right, you will hear a lot when we speak about genomics, this term called base pair, nucleotide, amino acid. This is where it comes from. So a single structure here is a base, and the connected are base pair. This molecule can be read from both sides. So you can see the 3' and the 5' ends. So you can read from either side. And this single base pair represents some kind of a nucleotide. So this nucleotide, we would refer to one of these adenine, guanine, thymine, and cytosine. So to understand how this code works and what is in this code, as you all know, this is the famous starting point of the Human Genome Project. You can see it was published in the New York Times on Tuesday, June 27, 2000, when they announced that this code of human life is cracked by scientists, right? So now scientists understand how these DNA sequences that we can now read are organized to inform us about life. What is life? How does life function? How does it produce the phenotype? Now, today, we are in 2021, 21 years later. So if that happened 21 years ago, what else do we need to learn about this? So it turns out that simply reading the code and annotating it is a much longer process than the human project itself. So the human project itself was focused on understanding what is in the code. And they discovered that there's only 1 to 2% of protein coding genes. The rest are non-protein coding genes. And so we have a lot of other types of information that is in the DNA. And we now know that there are a lot of questions, there are a lot of additional things to understand about the human genome, about the DNA, how it's organized, and how it actually works. And it's a very complex approach that people now take to study individual specific regions of the DNA and try to understand what is the impact of how this code is written to describe life and describe phenotype. So as we spoke, DNA is stored in the nucleus, organized in chromosomes, and specific regions are responsible for specific functions. Some of the easiest regions to read and understand their function are what we call protein coding genes. There's a total of 22,500 protein coding genes, but even that number continues to change. Protein coding genes are the sequences that are used to produce proteins, and they also have a specific structure. So when we think of this code working or producing that phenotype, the way it is done is by DNA being transcribed into RNA and then being translated into proteins. So these proteins that are produced from DNA is facilitated by several different proteins. It's a complex kind of a translational process that we will study in a little detail to understand exactly how it works. But the basic concept that we need to understand is that the individual nucleotides or base pairs that we saw in the DNA eventually have to be converted into amino acids, which is what the proteins are made up of. And so when we think of protein coding genes, we're going to be looking at sequences, dividing them up into tricodons, and then looking at how those trinucleotide codons, and looking at what they mean, how do they function in terms of amino acids. So the function of the RNA polymerase is to convert DNA to RNA, and then the ribosome, of course, converts the RNA into protein. So the genome composition, let's just talk a little bit about how the human genome is maybe different from other genomes. The human genome is not the longest genome. You can see right here's the human. Grape, for example, is much longer, not much longer, but longer nevertheless. And influenza, for example, flu or COVID, we all know is the thousands of nucleotides long, as opposed to the human, which really has tens of thousands of genes and billions of nucleotides. So influenza, for example, has 11 genes and is somewhere about 20,000 nucleotides, and that's how many genes we have. So here you can see what types of genes do we actually have. And you can see that 9%, so almost 10% of genes, we don't even know what they are. But how do we know that a gene is a gene? So a gene is also organized in a very specific way. They're not just sequences of nucleotides. They have upstream and downstream regulatory sequences that control transcription. Inside the gene sequence itself, there are codons and introns that undergo splicing before a gene is translated into a protein. And the length of these genes can vary significantly. And so they vary because they have these introns that could be very long. So when we speak about translation, like we said, the translation occurs as a result of ribosomes. So here you can see, this is a microscope image of unfolded DNA. You can see that the ribosomes are these large dots right here, and that is what they look like. So what happens is mRNA gets into the ribosome, and the outside of this ribosome, we already have this protein, which is a sequence that is folded and eventually forms a protein that looks like this. So this protein, this is actually two different proteins bound together because proteins now have a function. They interact with each other. And so in the context of genomic data analysis, we can look at every step of the way. We can look at the DNA. We can look at the impact of this DNA on transcription. And we can look at the impact of DNA in terms of translation. And so the ultimate goal of genomics is to integrate multiple aspects of this kind of data by looking at where the gene or genes are located in terms of the chromosome. What do we know about the organization of that gene and how that gene could be translated into a protein? So here you can see that protein structure. And eventually to validate its function through experiments. So here you can see this is the same protein. This protein is NM23H1. This is a protein that is responsible for control of metastasis or spreading in cancer. And you can see that its structure is known because we have a crystallized structure of the protein, but we also have studies that show how this protein functions, and we can measure this in terms of animal models, cell lines, and compare and contrast how it works in different variations. So when we talk about genomics in this program, we're going to be looking at data to try to understand what this data can tell us about a particular phenotype. And there are several different ways that we're going to be able to look at this. We're going to be looking at methods like multiple sequence alignment, variant calling, phylogenetic analysis. We'll talk about what types of mutations and variations we can find, which ones cause a change at gene expression level, which ones cause the change at the protein function level. So we'll talk about all of these things throughout this program. But the basis of this program is going to be based on data. And the data that we now study to represent genomic variation has evolved since the beginning of that human genome. And now we have thousands and thousands of human genomes, as well as genomes that are representative of pretty much a lot of organisms that you can study. So when you think of what you can do with genomic data analysis, once you know how to analyze it, you really have a huge potential of understanding, validation of specific types of variation that we can observe, including human disease and response to different treatments. And ultimately, this trend will continue. So knowing and acquiring the skills to analyze such data is a critical skill that you will take away with you to be able to contribute to the continuous exploration and understanding of how genomes work, what do they encode in terms of genes, and how can those genes be studied in terms of regulation of these complex conditions like cancer, like infectious diseases, like neuroscience, and others. So after you heard this brief introduction into the topic of genomics, I would like to pass it on to Dr. Raghav, who will speak about the other sessions in the program and kind of talk about more of the practical application of genomic data analysis in the context of several different projects. So Dr. Raghav, I'll pass it on to you.
Thank you. Thank you very much, Elia. Let me find ways to share my screen. I think you all can see my screen. So before continuing on the same level, let me introduce myself. I am Raghavendran. I am a research consultant and mentor with Pine Biotech and Omics Logic. I'm trained in bioinformatics and I hold a computational biology PhD from Max Planck Institute. So my research interest and focus is on data science techniques such as machine learning and deep learning applied to integration of multi-omics data like genomics data, like transcriptomics data, and like epigenomics data, etc. So my role in Pine Biotech includes developing curricula, mentoring students in the research fellowship program, etc. So for my research projects, I leverage a lot of publicly available data to infer very relevant biological research questions. So please let me talk about one such example in a very brief couple of slides, where I developed machine learning models to predict a tumor response for a variety of treatment options, employing multi-omics integration of transcriptomics data and genomics data and copy number variation data. So I don't need to introduce you to the adverse effects of cancer. As we all know, cancer is the leading cause of deaths worldwide. So for such a dreadful disease that we have known for a long time, the systematic cancer treatments have relied on using chemotherapeutic drugs that are basically marginally more toxic to cancer cells than normal cells. This is a huge problem because on an average, only 25% of the cancer patients respond positively to cancer drugs. But in comparison, about 80% of the patients respond positively to popular painkillers that are available in the market. So one prominent solution to this problem is to employ something technically known as precision oncological therapy, where a treatment strategy for cancers designed by targeting molecular characteristics of the patient's tumor. So hence, the treatment is personalized to the patients who are likely to respond to specific cancer therapies. there is not sufficiently enough data from cancer patients and not uniform data from cancer patients to molecularly characterize the tumors that we have under hand under multitude of different treatment options that is being currently studied in different clinical trial levels. So we have to resort to a suitable model systems. So one of the suitable model systems is the immunocompromised mouse model, which is also known as patient-derived xenografts. This is done by transferring the tumor cells from the cancer patients, and before transferring the tumor cells into the mouse models, we subject those tumor cells to next-gen sequencing techniques and gather information regarding genomic data, regarding transcriptomic data, regarding epigenomics data, and regarding copy number variation and other omics that are currently under study. So once these tumor grafts or tumor cells are transferred into mouse models, then these mouse models can be subjected to multiple treatment options or administered several cancer drugs that are currently under clinical trials and study, to understand the effect of these drugs and to understand the tumor's response to the respective drugs. So this study actually documents the response of breast cancer and colorectal cancer and other cancers to multitude of treatment options, and have published this research and let the data being available publicly. So this is done by Novartis, and the library and the data that they have deposited and is available freely is called Novartis Patient-Derived Xenografts Library or Encyclopedia. So this encyclopedia or this library has both the molecular characterization data of the tumor and treatment response that are available in the public repositories. So this is a huge amount of data, and so much of data, such a wealth of information in the data, generates a lot of research questions, and many research questions have explored this data for specific purposes. So my interest was to use this data to design and implement interpretable machine learning models to predict tumor response to different treatments. So in order to do this, since we have multiple biomarkers, or since we have multiple omics data available, so I had to employ a multi-omics integration at a different level. So generally, multi-omics integration can be applied at different levels, like as early as possible or as late as possible, depending upon the complexity and depending upon the appropriate strategy that will help us discover complementary information and maximize the information content available in this multi-omics data. So for this particular data, I had to include, or I had to integrate it at the late level because of the different distributions of the data along the multiple omics level profiling. So what did we find after using this multi-omics data to build a machine learning model? So before the analysis, I will just try to tell the accuracy metrics that I used to validate the prediction accuracy of the machine learning model that trained with this multi-omics data and tries to predict the tumor response based on a separate test data that is separately left over or separated. So the accuracy matrix is Matthews correlation coefficient, which goes from -1 to +1, where the +1 would describe an absolutely perfect prediction accuracy for the model, and -1 would describe a completely negative or completely inaccurate prediction accuracy of the model, and anywhere between 0 is going to describe a random prediction accuracy for the machine learning model that I have developed. So traditionally, for these questions or for understanding the treatment responses, people have looked into the single gene biomarkers that characterizes the onset of cancer, and they have tried to find out whether these single gene biomarkers can also be employed to predict treatment response. But we found out this is not a very accurate model, as you can see from the graph. When I use the traditional single gene biomarkers that characterizes this breast cancer level at different from different different publication for these different treatment options, I see that their prediction accuracies are not very great. But the moment that I include not single gene biomarkers, but information from all of the genes, genomic information from all of the genes, I see that the prediction accuracy slowly start to get better. And I also see that from the precision of the prediction accuracy that is plotted for all the treatment options that is given in this box plot. So the availability of multi-omics generally requires, as I mentioned, very complex integration of this data to make sense. So here, increasing the information content by including multiple omics data, we can see that the accuracy of the machine learning model that trained from this data generally increased towards, or everything is going towards +1, and everything has a lot of very good precision in terms of their prediction spread. So this clearly tells us that there is a lot of promise in using this multi-omics data to predict the treatment response using these mouse model systems. 1, 2. This also tells us by increasing the information content and by including not a single gene instead of single gene markers, by including many more genomic information and many more multi-omics information, we can increase the accuracy of the machine learning model that predicts the tumor response by learning from the distribution of this machine learning from this multi-omics data. And 3, we can probably extend this analysis to a patient cohort or patient information that we can download from TCGA repository, just to test, just to investigate how a model that is developed and that is trained using multi-omics data that is generated from mouse models can or respond to patient data from TCGA repository. So what more could be done? I mean, from these results, we obtained a list of informative genes that are currently being investigated for identifying specific biomarker set of genes that can define, or that can be inferred in a mechanistic detail about the tumor response to different treatment levels. So these research projects and these questions can be can be performed by using publicly available data and by using methodologies that are not completely different from what you are going to learn throughout this program and throughout the process of and throughout the lessons that are covered in LearnOmics platform. So we using the resources in Omics Logic platform will demonstrate you how to perform such powerful, impactful research and by using or by accessing publicly available data. So as you have been briefed before, the resources in this platform, in the Omics Logic platform, and in this program, in this genomic program, is aimed at introducing you to omics data and their relevant analysis. And this will eventually help you create or understand or ask a highly impactful biological research question that you can start to explore on your own. So we will demonstrate you over the course of this program how to perform such high quality analysis on genomics data. We will also show you how example projects that we have developed, which start from the first base of formulating a hypothesis or exploring a data, designing appropriate analysis methods, and to perform complex analysis, and in the end, interpret the results in a biologically meaningful way. So as you have all been briefed about the NGS generate large volume of raw sequence data. So to analyze such a raw data, many different tools for genomic data analysis are used. And these include alignment of reads and full sequences, analysis of relationships between these sequences and their evolutionary divergence, measurements of the gene expression and their relationships between sequences, and the relationships between structures of protein that these gene sequences encode to, as well as variations or variants in the gene sequences and the sequences of the genome that can be termed as somatic or germline variants using multiple different calling methods. The Taubot Bioinfo platform is an example of a big data platform that can be used or that can be leveraged to perform all these analysis and many others using an intuitively user-friendly way. So when the analysis is performed using a raw data, which is usually stored in FASTQ file format, there are several important steps to consider, such as pre-processing, mapping, variant calling, annotation, and other associated steps also. So for all these steps and each and every step, we have at our hand a multitude of algorithms that are available, which does basically a similar job, but with a slightly different manner and slightly different protocol. So in this program, we will teach you, or we will help you learn how to use this pipeline and how to use different algorithms that will suit the data or that will suit the question that we have currently in our hand. So for each task, you can connect the analysis jobs into pipeline, and each one of these analysis pipeline will include various inputs and will have various outputs that you can download and use it for downstream analysis. Sometimes for the outputs, it is just sufficient to visualize the FASTQ alignment files to a reference genome sequence to quickly get a sense of major variants, or to quickly get a sense of how our alignment varies across the reference genome, and what are the different variants that we can observe and that we can find that are distributed along the different genes and different parts of the genome. So this is specifically true, or this is especially true when you have a short sequence, or when you have a targeted regions that are sequenced, and these are very easy to explore. But however, to explore a long whole genome sequences and to visualize that and to visually interpret that are extremely difficult. So a computational interpretation and a computational analysis is performed in addition to the visualization that we can use, where this computational analysis can call the differences in the FASTQ read files to the reference genome. These are called variants, and these variants are called from many reads. And in the case of somatic mutations in cancer, that might include downstream annotation of found variants based on the reference database. So these variants can include mutations of different kind, and for each one of these kind, you will have explanation or how to interpret those different mutations and variants that are explained in the following sessions in this program. So to visualize the alignment of the FASTQ files to the reference genome, we will use open source applications, and we will use Taubot Bioinfo platform also to visualize and to learn about their mapping onto the different parts of the genome. So large peaks, I mean, this is the reference genome that is in the gray area, and where it has been so far zoomed out that we cannot read the individual nucleotides as you have seen that constitutes this genome. And these gray large peaks indicate high coverage in these areas, and shallow peaks will indicate low coverage or low confidence in alignment, as you can see straight away from the gaps in the reads that did not cover these areas. And these vertical lines in purple or in pink lines will tell you what are the differences that we observe at different points in the in our sequence that are aligned to this reference and with respect to the reference. So such visual representation and such visual analysis, as I mentioned, might be sufficient for genomes of small sequences or of targeted sequence experiments. But when you are going to perform such an analysis for 3.2 billion base pairs, it is going to be extremely exhausting and is going to be very heavy for the computer to also work. So that's why multiple downstream variant calling methods and multiple downstream analysis methods are employed to analytically quantify the variants we observe in our mapped sequence with respect to the reference sequence. So a zoomed in version of these variations or these visualizations often include visualizing in terms of sequence alignment. So these sequence alignment can refer to excuse me, two types of alignment. The one it can refer or it can be aligned to a reference, or which is called a mapping, such as when you have a FASTQ files, as I have mentioned, from a sample, and you wish to align each read to the best position on the genome to find whether these FASTQ files or whether the sequences in the read or the multiple reads reveal a variation from the particular reference. And in that case, or in another case, multiple sequence alignment, this is when you have multiple consensus sequence that you would like to compare with each other and to develop an understanding of similarity among them or between them. So these both of these alignment can also be performed in Turbot Bioinfo platform. In many cases, the multiple sequence alignment can be or is used to find structurally important variation, and such an alignment or such an analysis can also be performed with amino acid protein sequences that can be obtained or that can be elucidated by just simply translating the gene sequences into their respective amino acid coding sequences. This is done by taking the trinucleotide codons, as Elia has mentioned, and then translating them into their respective amino acid sequences. So the differences in both at the genome level and specifically at the amino acid level can often be visualized and can often be identified using or can often be visualized on the three-dimensional structure of the protein that is encoded by the gene, and the location of this mutation on the protein structure and their effect on the function of this protein structure will enable us to make meaningful interpretations of the variants we identified as the result of genomic analysis pipeline. So in another way one can use genomic analysis is by carrying out what usually is called a genome-wide association studies. So to carry out a GWAS or genome-wide association study, researchers use two groups of participants, people with disease being studied and similar to people without the disease. Sometimes this can also include certain variations of the disease or certain conditions that are being studied to identify those genomic areas that are responsible for this altered state or conditions. So researchers then obtain the DNA from each participant, sequence their whole genome, and then if certain genetic variations are found to be significantly more frequent in people with the disease compared to the people without the disease, as you can find in some of these plots here, they can often be associated, or these variations are said to be associated with the disease. These associated genetic variations can serve as a powerful tool or powerful pointers to those regions in human genome where the disease causing problem might reside, or that can be interpreted as very relevant regions that are associated with the disease or the altered state that is currently under study. So the downstream analysis of these found mutations, variations, these SNPs, and often these can be represented in or presented in a very different way, depending upon the purpose of analysis or depending upon the purpose of the use case. I mean, it can at its simplest form can be represented as a table, and at its more graphical form it can be represented as a lollipop plot and along the gene sequence, or it can be visualized on the three-dimensional structure of the protein encoded by the gene of interest, and then it can also be visualized as a it can be visualized using multiple different visual forms, and can be visualized along the pathways to which multiple genes that are associated with, and how these different mutations or these different variations in these genes might affect the pathway that involve these genes that are currently under study. So this introductory course leads into multiple courses and projects that can serve as a springboard into the world of bioinformatics for all of you. The curriculum for this introduction to bioinformatics, the curriculum for this genomics course is carefully planned to cover a broad overview of different types of methods to analyze the genomics data, and also to understand how these variety of methods are used to answer very relevant biological questions that are found in research papers involving genomic data and its interpretation. So to learn more about these analysis methods and their applications in various project examples, you will often be referred to associated lessons in the genomics course series and also in other courses in the Learn Omics Logic platform. These lessons will include more expanded overview of the key concepts, principles, and examples of genomic data analysis. Excuse me. These online courses cover, or these online coursework on our education portal will provide you with introductory materials that have been used multiple times by thousands and thousands of people from all over the world, and they have found these perspectives very exciting. And each of these courses in the end also contains quizzes and also contains assignments that will help you, or that will allow you to see how much you have learned from this course, or throughout your progress through these different courses and different lessons. So in addition to these courses, by seeing how experts analyze their data and how these impactful research results are achieved, you can start developing a research question on your own to identify appropriate type of data and planning and also to design your analysis plan. So for this purpose, we have in our platform have carefully prepared some exemplar projects that will use all the skills and all the knowledge that you have learned to infer highly impactful biological relevance and to answer high quality research questions. So these projects include a variety of data, a variety of analysis methods, and ask different questions that are related to using genomics data and and to using multiple different analysis methods to infer high quality biological interpretations. So these practical exercises and exposure to data driven discovery using real research data will encourage a critical thinking for most of you. And the purpose of doing a practical assignment and the purpose of going through these projects that we have developed and then learning along with the projects that we have developed will help you gain a deep understanding of these multiple genomics concepts and the processing and the analysis pipelines involved with it, as well as you can also showcase how various methods of analysis are applied in real world situations. So in the end, by following through these example projects and by also going over the course 9, which will help you in designing your own research projects based on the theme of genomics data, or based on the theme of based on the research area of interest, you can actually perform, or you can perform a project on your own. You can perform a research project, you can answer a research question on your own, and you can have your own research experience that is satisfying satisfying for applying various skills that you have learned in this course so far. So that will conclude my presentation. I will take some questions if you have any, and if you have not, I can hand it over back to Sonalika. Okay, I have specific questions. Using multi-omics data, how can you exclude experimental errors? I mean, yeah, if you use multi-omics data, we can actually normalize these multi-omics information data using z-score normalization, and we can probably look for the data distribution by different normalization methods and different log transformation methods, and then maybe we can exclude experimental errors. In FastQC, when FastQC2, we can perform the annotation. You will have your answers. You will have how to analyze FastQ files and FastQC quality check, how to interpret the results in the further sessions in this genomics lessons, Harish Chandra. Okay, I will hand it over now to Sparsh, and then we can go ahead with the session we have at our hand.
Thank you so much,
Over
Dr.
to you,
Raghavendra. Thank you, sir. Now I would like to share my screen once again and take you all through the registration steps. So this is for them, those who have not yet registered for the program. And Suganya will be able to share with you the recording. Also, we are live on YouTube right now, so you can follow the link and go through the session from the beginning, and let us know if you have any questions, and we'll be able to assist you with the same. Okay, So this is the program page. Again, back to it for the steps to register yourself for the program. Now, once you are on the program page, for those who would like to register themselves for the program, the first session of the program was an open session for everyone, for the interested candidates as well, so that you could join and see how this online training program is going to work. And from the second session onwards, the enrollments will be able to join and proceed further. So when you scroll down on the program page, you'll find three subscription levels. The first one is a beginner one, the second is intermediate, and the third one says advanced. Now, these three subscription levels are going to give you the access to the various resources and different resources. In the beginner subscription level, you will be able to be a part of the live group sessions. In the intermediate one, you'll also get one-on-one mentor support. And the advanced subscription level will give you some extra time a month to work on your research project. So after the program gets completed, after the particular training program, you will be proposing your research question, and then you will be working on a research problem with the mentor. So these are the three different subscription levels that you can pick based on your requirement and get started with the genomics data analysis program. If you have any questions, please put them in the chat box so that we can answer. And I'm also putting in our mail address, that is marketing@omixlogic.com. If you have any questions, please reach out to us on our mail address and we'll be able to assist you accordingly. Also, please make sure that you have added this mail address to your address book, because many a times the mails drop in the promotions tab because of a lot of interactions taking place between us and the interested program participants. Also, I'll like to share one more insight for the people who have registered themselves for the program. How are we going to communicate? The answer is Slack. So this is how the Slack channel looks like, and the registrant participants would have received an invitation mail from me on the Slack channel group, for the Slack channel group on their Gmail accounts. So please accept that invitation, and accepting that invitation will redirect you to this group. And in this group, I'm going to post in the session details, the meeting IDs. Also, I'll be posting in the session recordings after each session commences. And also, we have a mentor here in the group itself. So you'll be able to take your questions in the group here, and the mentors will be able to assist you accordingly. It is Slack, Suganya. Only the program and participants will be able to join this particular group, and I've sent out the invitation. Suganya, I'm not sure if you have received your registration, so please reach out to marketing@omixlogic.com and they'll help you with the Slack channel links for you to join. Thank you, Suganya. And do we have any other questions? If not, then I believe we are good to go. And we'll be now meeting in the second session. Okay, so I'm taking that as no, that we have no other questions. So it was very nice to have you all with us for the first session, and looking forward to meet you all in the second session. Now I'll just share the recording of the first session with you all so that you can go through the concepts if you missed any.